Structures of proteins. So you have 4 sections in this. A protein has a primary structure. A protein has a secondary structure. A protein has a tertiary structure. And also in the 4th one, quaternary structure. So on the top you have one structure, then one another inside it, and one another inside it, one another inside it. I will explain this section using one example. So suppose you have a protein. Suppose you know that the primary structure of protein is a linear chain. You know the primary structure of is a linear chain. So the protein looks like this. It is a linear chain. When you further analyze it, you see that the linear chain, when you see it closely, you see that the linear chain is actually curved, and it is in its. So on further analysis, the first one, from far away you saw it as a linear chain. It was linear. When you see it further, it was spiral. When you see the spiral closely, you see that the spiral was also not all straight, it was curved. It had bends in it. So it was something like this. And the 4th structure is that you find that this whole structure was not formed by a single chain. There were 2 or more chains in it. So you saw that there was one chain going like this, and you also saw one chain going like this, and these 2 chains attached to each other. So I'll just explain these 4 sections like this. In the case of first, the specific sequence in this, the various alpha amino acids in the protein are linked to one another. So over here in the first section, we only analyze it based on the which alpha amino acid is attached in which sequence. So over here we only talk about the sequence of alpha amino acids, that's it. So for example, if you take a normal hemoglobin, in a normal the sequence is like first is val, then valine, histidine, leucine, threonine, proline, GLU, then lysine. I have to mark this one. Okay. Then there is a disease which is known as sickle cell. If you might have heard, sickle cell hemoglobin. Okay, so you know one thing, that a normal hemoglobin looks like a disc shape. It looks like a disc. Whereas there are in some of the blood, we see that the hemoglobin is not like a disc. It is like a sickle. It is bent in a C-shaped. So in this, if we see closely, they have the sequence is like valine, histidine, leucine, threonine, proline. Then over here, where you had to see glutamic acid, in this place you have a valine. Then the same is it. So just if you just change one single acid, the overall structure changes. So in the primary we only see that how are the alpha amino acids arranged with each other. That's it. If the arrangement has changed, the shape will change and all the other properties will change. Okay. So now if I know the how it looks like, the second one is come, how is it arranged? So the so it says that so in this what happens is that the different chains are attached with each other using hydrogen bonding. Hydrogen bonding. They either contain hydrogen bonds or they either contain disulfide bonds. So these series are formed like this like 1 2 3 4, and they are attached to each other by hydrogen bonds. Now under this section you have two kinds of structures. The first one is a alpha helix structure. So you have to make strands. So either the strands, the chains you formed in the first one, they are either coiled around the axis like this to form an alpha helix. By the term alpha helix, I mean that the chain is right-handed coiled. If so, you have to apply the rule of right hand. So it has to go like this. Use your right hand thumb rule. So example of alpha helix kind is your nail wool. Second type of structure it might also happen is that these chains, these patterns were not coiled, they are placed to each other, parallel to each other. They were joined using hydrogen bonds to form a surface. So this kind of a system was called beta pleated sheet structure. So they are stacked to each other. Now under this section you have two different types of sheets. You don't have to remember it, but just for knowledge, you either have a parallel sheet or you also have a anti-parallel sheet. You don't have to remember this. Just remember this: the strands you formed in the first section can either be joined to each other and may have a helix structure, or they may have a sheet like structure. Now these sheets are stacked one over the other like this. So this sheet looks like a paper and the stack looks like a book. Simple. Now comes a third type of structure. So what happens is that, so over here you formed an alpha helix, over here you form sheets. Now this alpha helix is not straight, all straight. These alpha helix may also have some bends. It might be bended like this. So these kind of bends are seen over here, or these sheets might have a U-shaped. So what happens is that in the fibrous structures, the structures having fibers, in the fibrous structure they have a tertiary shape of rope-like. They are joined to each other by H bonds. So rope-like for fibrous. And the second one, if you have a tubular protein, they form the entire molecule of shape of sphere. Okay, so the third one shows only how much is the angle, how much is the bend in it. Okay, in the fourth one, what we see is that what happens is that sometimes the proteins do not exist as a single chain. So some of the proteins exist as subunits, or these units are also known as protomers. Okay, so you have to analyze these subunits, the shape of these units. So once you get all these four structures, you finally know how it looks like. So in short, I can say that the structure of protein is complex. You have to analyze it from every angle possible.
Thank you for watching. Visit us at needbook.in.